Hi everyone, and welcome to our poster. In this work, we develop techniques to assess the relative solvent accessible surface area. It's a well-studied problem. However, to the best of our knowledge, we are the first group to use graph neural networks to solve it. The best way to describe our approach is to first look at the input and the output. We take a dataset from COSP11. For each protein in this dataset, we generate edge and node features that will be fed into our network. As node features, we use one-hot encoded sequence and PSSM, or positional specific score matrix, that we generate from multiple sequence alignment of the homologs. As edge features, we use the distance matrix. This matrix can be inferred from the alignment as well. As for the architecture, we built our network using building blocks of the protein solver model that was published last year. The output is a value from 0 to 1 for each residue in a given protein, the relative solvent accessible surface area. We achieve very nice results, and on this figure, figure 2, you see per residue predicted values of accessibility and how they correspond to the true values inferred from a PDB structure. On this figure, figure 3, for a different protein, you see how nicely predicted values correspond to true values. But these are individual examples. Overall, on the test set, we achieve 8.5% of absolute error and almost 80% of Pearson correlation. We also test our model on the independent Manege dataset, which has been a popular benchmark in the field. And after removing any overlaps with the trained data, the performance on this dataset is almost the same. We do have some outliers. They're highlighted here in red. But as this plot shows, the majority of them are just short peptides. Therefore, it might be challenging for the network to solve them. I need to emphasize that it's a work in progress, but we already outperform all previously reported classifiers. The ability to have such great accuracy and accessibility makes our network, our approach, a practical tool for the drug discovery pipeline, where a solvent accessible surface area is an important factor when targeting a protein. Our graph neural network can also be trained to predict the secondary structure and/or accessibility simultaneously. This will be the direction of our future work. Thank you for your attention.